Today we are going to connect the upstream and downstream stories of the endocrine system. In the endocrine system, the upstream story is the glands. And one of the things we have to discuss with the glands is the kind of complicated story of how hormones are released. When do we decide to release hormones from the endocrine glands? Now, the downstream story is the story of targets and receptors. And this is downstream, and this is going to be where you're going to be receiving signals, so you have to have receptors there. And one of these terms that we're going to be emphasizing today is the term response. So release happens at the end at the upstream end, and response happens at the downstream end. So we're going to do some quick and dirty sketches of our endocrine glands themselves. The pineal gland is in the diencephalon of the brain. And then just inferior to the brain, we have the pituitary gland with its infundibulum there, the pituitary. There's actually two parts to the pituitary, so one is a gland and one is not, so we'll just call it the pituitary. Obviously not to scale and not in the right position in the body, but here we have our thyroid gland, which wraps around the trachea or the windpipe. And we're going to be specifically discussing the structures on the posterior side of the pituitary gland, which we're going to be drawing in dotted lines to indicate that they are on the posterior side. These are the parathyroid glands, and there's between 3 and 10 of them on a thyroid gland. The parathyroid glands. Next, we're going to draw the thymus. The thymus is only active about the first 2 years of life, and it is anterior to the heart. Below that, we're going to go ahead and draw our pancreas. This kind of yellowish, almost fatty, glandular-looking structure, sort of an elongated triangle. The pancreas sort of obscures 2 of the structures we're going to be discussing in a bit. The adrenal glands, the adrenals. Now the adrenals rest on top of the kidneys. That's how they get their name, on top of the kidneys, adrenals. So we're going to outline some kidneys there as well. There's 2 structures on the adrenal glands that we're interested in. We're interested in the adrenal medulla, which we actually discussed in the last unit. And we're also interested in the adrenal cortex. The medulla is in the middle, and the cortex is along the outside. The last of our specific endocrine structures we're going to be drawing are going to be the reproductive organs, the gonads, and so I'm drawing, and a uterus that's in trouble, man. Anyway, there it is. There's my uterus. But anyway, so in between the ampulla of the uterus, we have 2 oval shapes here. Those are the ovaries. These are important for production of certain sexual hormones. And so the ovaries are the female reproductive, and on the male reproductive side, we have the testes, which are wrapped around with something that looks very similar, which is the epididymis. All right, so we're going to begin our story of how we decide when a hormone is going to be released. You can't arbitrarily release hormones all the time. You have to only allow hormones to be released from endocrine glands under certain physiological conditions. And so these are the 3 conditions. There could be a humoral stimulus, there could be a neural stimulus, or there could be a hormonal stimulus. So let's go ahead and discuss our humoral stimulus first. The term humor refers to the blood, and in this case we're going to be looking at blood calcium levels, which are supposed to be maintained at a particular level. When your blood calcium levels get too low, there's a blood supply that goes right through those parathyroid glands that in turn causes the parathyroid glands to release parathyroid hormone, or PTH. Now when PTH is released into the blood supply, which we're going to be drawing in a moment, they're eventually going to be targeting osteoclasts. They actually have 3 different targets, but one of their targets is going to be osteoclasts. And the specific response of these cells, it's important to make the connection between the release of the hormone and the response of this target cell. The response in this case is going to be enhancing the production of the osteoclasts in this case, which would be increased acids and enzymes released, which in turn eats away bone material and causes the release of calcium back into the blood and therefore raises the blood calcium levels. All right, so let's go on down to the neural stimulus here. So the neural stimulus, we discussed this one last week. The adrenal medulla is stimulated by the sympathetic nervous system. And so these neurons will plunge right into that adrenal medulla and will cause those cells within the adrenal medulla to release epinephrine and norepinephrine. And there's many, many targets of epinephrine and norepinephrine, but the one we discussed last week were the heart cells. We're going to go a little bit more in detail here and just say that the response here would be to enhance sodium influx, which in turn makes the cells more excited. We're not going to be continuing that story today, but we are going to be continuing that story in a couple of weeks when we talk about the cardiovascular system. And lastly, when we discuss hormonal stimulus of hormone release, we are discussing the idea that a particular hormone can come along, which would be called a tropic hormone or a maintenance hormone will come along and will cause the release of other hormones. Oftentimes these tropic hormones, these managing hormones, will be released by the anterior pituitary gland. And this is one example. ACTH, it's called adrenocorticotropic hormone, and it is the tropic hormone that causes the release of several hormones actually from the adrenal glands, and particularly from the adrenal cortex. And the one we're going to be discussing today is going to be cortisol, which it's got a lot of things going on with it. It's a glucocorticoid, but I would say it's best known as your stress hormone. So therefore it has many, many targets all over the body. There are many targets of cortisol. Many cells of your body have glucocorticoid receptors or cortical receptors, but the one we're going to be discussing and focusing on are going to be muscle cells, because there's a particular specific response that muscle cells have, especially to elevated levels of cortisol, and that would be to increase their production of a protein called ubiquitin, which over time begins to break down the proteins inside of muscle cells. Let's go ahead and use our red marker to indicate a blood supply. And in this case, we're going to label it as upstream and downstream. So again, the upstream signal is going to be having to do with the glands and what it is that they're releasing and how it is they're releasing those things. And then downstream is going to be target cells and receptors. So we're going to go ahead and pop the 3 hormones that we've produced over here into our bloodstream. To give you a little visual on this, PTH, we're going to make little circles. We're not actually going to follow the story of epinephrine, but we do pop it in there just so you know that it's of course in there. And cortisol, we're going to have this little kind of half moon sort of a thing going on. So we have our PTH, our epinephrine, and our cortisol in the bloodstream. So now we're going to be turning our attention to what happens next. What happens downstream of this when the hormones leave the bloodstream and enter into the interstitial fluid and start to interact with all those vascular tissues all over the body? Who do they actually interact with? So one of the things we can do then is we can draw 2 different hypothetical cells. Maybe they're right next to each other, could be. And we're going to draw 2 different receptors on their surface. The first type of receptor has a little divot that is round, and the second receptor has a divot that is more of a V shape. And then we have this hormone that comes along, and which of these cells is the hormone actually going to interact with? It's going to interact with the receptor that actually fits it, and so it's going to have that receptor that has the divot instead of the roundy. So that makes because the receptors fit that hormone, that makes those cells, the one that's portrayed on the right in this picture, that is the target cell. So what makes a target cell is that it has the correct receptors that fit, like a key fitting into a lock, that particular hormone. That's it. And it doesn't matter how much hormone you have pouring out of the bloodstream and pouring into the interstitial fluid, that non-target cell will never respond to that hormone because it does not have the correct receptors for it. Only the target cells, only the cells with the correct receptors will actually have a response. Now, let's talk about two different hypothetical cells that both have a receptor for the same hormone. They're both sensitive to the same hormone. They will both have a response. But in this case, the cell on the left is going to have many receptors for the hormone, and the cell on the right is going to have fewer. So let us say that this is actually the same cell, but in different points in time over the course of a person's life, for example. On the left-hand side, what we would say is that this person has upregulated the receptors for this particular hormone. And on the right-hand side, we would say they have downregulated the receptors. A simple example of this would be growth hormone, which targets the growth plate inside of the long bones, but only for about the first 18 to 20 years of a person's life. After you've reached basically your full height, you no longer have to lengthen the long bones, and so you no longer have to target the growth plate of the bones, which are actually not directly targeted by growth hormone. There's an intermediary in there, but the basic idea is that they're no longer sensitive or no longer as sensitive as you get older to this particular hormone. Growth hormone does tend to affect changes in other cells throughout a person's lifetime, but it no longer causes lengthening of the bone after a certain point. So what we're saying is that these cells inside of the growth plates, inside of the epiphyseal plates of the long bones, is sensitive to growth hormone or to its intermediary during the first 2 decades of a person's life, and therefore has many, many receptors for this. But later on, you start to downregulate these receptors because you no longer need to be as sensitive to it. You no longer have to have that same response that you did previously. So growth hormone continues to be produced, but will no longer affect changes in these particular cells at certain stages of life. And so there's many, many different implications to this story. That's one simple example, but there's many examples of how this works. You can change the receptor count basically on the outside of the cell simply by transcribing more genes, more of the gene for that particular receptor, or transcribing fewer proteins out of the gene for that particular receptor. Transcription of the gene can be increased or decreased, and so that's what we call upregulation and downregulation. All right, so now that we've kind of discussed the concept of the target cells, we now have to classify our hormones based on which type of a receptor they actually interact with in the target cells. And so the first class of hormone we're going to look at is the amino acid hormones. The amino acid hormones are polar, and almost all of the hormones that we discuss are actually amino acid hormones. They use a system called the second messenger system, or a G protein mediated system. And so let's discuss that. Now, because the amino acid hormones are polar, they must remain outside of the cell. So we're going to go ahead and draw a cell membrane here. And the first thing we're going to draw is a transmembrane protein. You see, it's a protein. You may immediately think to yourself, it's probably a receptor because you see that kind of characteristic divot on the outside, but you also see a divot on the inside of the cell. And speaking of outside and inside, let's go ahead and mark our interstitial fluid, which indicates the outside of the cell, and our intracellular fluid, or our cytoplasm, which indicates the inside of the cell. The outside is portrayed above, the inside below. Okay, we have our PTH that comes along. We're continuing this story. Our PTH comes along and interacts with this receptor, this transmembrane receptor. It fits into that divot there. In response, the receptor is going to send a signal into the cell to interact with this G protein. Now you'll notice, of course, that the PTH, being polar, cannot pass through the bilipid membrane and enter the cell, but it can still effect changes within the cell. So this G protein is activated by GTP, which sounds a lot like ATP and is actually pretty similar. So GTP is hydrolyzed into GDP and gives off a burst of energy, which in turn changes the configuration of our G protein. And there it is. The G protein changes its configuration, moves down the length of the membrane, actually just a little bit down the length of the membrane. The activated GTP then, the activated G protein will then interact with this enzyme called adenylate cyclase. And go ahead and draw that GTP into that little divot spot there. And we'll go ahead and continue the membrane a little bit. The adenylate cyclase does something quite important. It mediates the conversion of ATP, which has been known up till this point as the energy currency of the cell, but it does something else too. It's able to be converted into this very important structure called cyclic AMP, or CAMP. Now, cyclic AMP is able to interact with, for example, protein kinases that are inactive, and it activates them. So we're going to be showing this conversion of inactive protein kinase into an active protein kinase. And that activated protein kinase can then go on to do a number of different things inside of the cell, including the thing that we are focused on, which is activating some kind of a response inside of the cell. And in this case, we indicated, well, what is the cell we're actually dealing with here? The cell we're dealing with is an osteoclast. And so the response of the cell here is to increase the production of acids and enzymes from this osteoclast. This in turn will start to eat away more bone material, which in turn will release more calcium into the bloodstream, which will raise our calcium levels, which was the response that we were really after in the first place. Now, our first class of hormones was amino acid hormones. Our second class of hormones is the steroid hormones, which are distinctive because they are non-polar, or they are fatty, and therefore they are able to actually pass right through that plasma membrane and use a very powerful mechanism called gene activation to get a response from their target cells. So let's go ahead and draw a cell membrane. These are phosphate heads on the outside and fatty acid tails in the middle. And because the fatty acid tails are the bulk of the cell membrane and they are nonpolar, only other nonpolar things can actually pass through them. So let's go ahead and draw our steroid hormone, which in this case is cortisol. And we drew it the same way as we drew it passing through the bloodstream there, a little half moon. Now it's going to pass through the bloodstream and it's immediately going to interact with its receptor. So this is an internal receptor. And these two together are going to form a receptor hormone complex. This receptor hormone complex is now basically activated and is able to go on and do other things. Namely, the receptor hormone complex, this steroid hormone called cortisol interacting with its receptor, is actually going to be able to pass directly into the nucleus of this cell. The nucleus is renowned as a structure that holds the DNA. So here we are inside of the nucleus. We've passed through the nuclear pore. And now we're going to show a strip of DNA there, a little double helix of DNA. The receptor hormone complex is going to actually interact directly with the DNA or with RNA polymerases that then cause the next thing to happen. So they're going to interact directly with the DNA to cause the transcription of a particular gene of interest, to cause the production of an mRNA transcript. The mRNA transcript then leaves out of the nucleus via a different nuclear pore and re-enters the cytoplasm of the cell. And now it's going to interact with ribosomes. Remember this whole story? It's going to interact with ribosomes and they're going to undergo translation. So many different ribosomes will interact with one single mRNA transcript. And so we go back to our DNA. We say, well, DNA is transcribed into mRNA, and mRNA is translated into amino acid sequences. So we're going to go ahead in brown there, we're going to draw some amino acid sequences, which will eventually fold their way into a protein. And the specific protein we're looking at, of course, is ubiquitin. If we looked back at our response that we had drawn and predicted in the first place, it was to enhance the production of ubiquitin, which eventually over time is actually going to break down other proteins inside of this muscle cell. So the presence of cortisol in this case can actually be eventually actually harmful to cells. They can start to break down their actual cellular activity. Well, there was a lot of information covered here, so I wanted to give a good review of these two different stories that we followed from beginning to end. So let's go back to the original story of the parathyroid glands. The parathyroid glands receive a humoral stimulus, which in this case is blood calcium levels. When the blood calcium levels are a little bit low, this stimulates the parathyroid glands to release a hormone called PTH, or parathyroid hormone, which in turn enters into the bloodstream and eventually leaves the bloodstream and enters the interstitial fluid surrounding a number of different cells, and some of those cells will be target cells, such as the osteoclasts. So we're looking at the cell membrane of an osteoclast here, and there are some receptors on the outside of it, such as this transmembrane receptor, which when it receives the signal from PTH, there's a number of cellular mechanisms that in turn, GTP, adenylate cyclase, cyclic AMP, activated protein kinase. Eventually you have a cellular response, which in this case is an increase in the production of acids and enzymes. Additionally, it should be said that it is very likely that you will start to simply create more osteoclasts in the presence of PTH, especially over time. So now we're going to follow the other story that we followed from beginning to end. We're going to go back to the story of cortisol. So here we have hormonal control of hormone release, hormonal stimulation in the form of ACTH, which was originally secreted by the anterior pituitary gland, by the way. We'll look at that more in the next lecture. The ACTH then interacts with the adrenal cortex to produce cortisol, which is a glucocorticoid. The cortisol then enters into the bloodstream and eventually leaves the bloodstream and enters into the interstitial fluid surrounding a particular cell. In this case, it is a muscle cell. And in this case, because cortisol is nonpolar or fatty, it's able to pass directly through that cell membrane, interact with a cytoplasm-based receptor, and forming a receptor hormone complex. This receptor hormone complex then can pass directly through a nuclear pore into the nucleus, interact directly with DNA, causing the transcription of an mRNA transcript, which then is translated into proteins in the cytoplasm of the cell. It leaves the nucleus and goes into the cytoplasm, interacts with ribosomes, and ends up forming more and more of a particular protein. In this case, the protein that is formed is ubiquitin. So the purpose of this lecture was to ensure that we were able to make a connection between why is it that hormones ever are released from endocrine glands, and how do they ever interact with target cells, and why. We wanted to tell a couple of examples of the whole story. I know there's a lot of details here, but there are lots and lots of details always to be known in the endocrine system. And my job, I feel, is to make connections in those stories. So hopefully this was beneficial to you.